Okay, good morning everybody. My name is Mikkonen, and yeah, I come from the University of Jyväskylä. I'm working there as a postdoctoral researcher, and I'm an environmental microbiologist. I'm actually curious to hear a little bit about your backgrounds. How many of you have your background in the wet lab, in like pipetting and yeah? Maybe more than half. So a similar kind of background as I do. How many of you have your background in computer classes, and so like real bioinformaticians, or with that background maybe? About 5, 6 of you? 7? Good. I will be talking from my background. So if and when there are things that like I don't explain very well, then please just stop me at any time and ask, like, what do you mean by this? I would very much like this morning session to be interactive. So, you know, you are an audience full of experts, and it's very likely that some of you know some of these things better than I do. So please, like, feel free to like jump in and give comments and ask questions, and let's make this interactive, because I have, I think, 36 or 37 slides, and if we don't make this interactive at all, it will be a little bit boring. Yeah, so environmental microbiologist, definitely background very heavily in experiments in the wet lab. And during the past few years, I have been working with Illumina data and with ion torrent data. And also partly because of my background, because of lots of experiments and lots of things that have gone wrong, I hope I will be able to give you examples of like recommendations of what do and what not to do, like how to avoid the mistakes that probably most of us do at some point. So this will be kind of an introduction into community analysis by Amplicon sequencing, mostly by 16S sequencing. So first, I would just like to remind you, maybe probably most of you already know, but just to remind you what this kind of microbiome analysis or Amplicon sequencing based community characterization actually is, what it tells you and what it doesn't tell you. I will also tell you about the effect of PCR conditions on the figure of the community that you recover. And I will also be telling you about the effect of different sequencing platforms. So mostly comparison between MySeq and ion torrent PGM. If everything else in my talk is very familiar with you, if you already know everything else, then at least here I hope I will be able to give you something new, because I got my results with the very newest chemistries last week, so at least those you haven't yet heard about. I will also be telling about a little bit about different analysis platforms, so about mothur that I'm most familiar with, a little bit about other platforms and other calculations and kind of like known issues or known problems with different platforms. And this is basically what I hope you to remember or understand after these about 35 more slides. So if you don't know exactly what you're doing, just do what everybody else is doing, because then you can justify it in a publication. If there are already 20 publications that have done the same thing, then you are pretty well off justifying your choices to the reviewers, because of course you want to publish your data. And like, I know that's kind of skeptical, but nowadays very often when I think like what to do, I wonder, can I justify this to the reviewers that will read my paper? Yeah. The second point is that if you want to compare your samples, you really need to do everything, and I mean everything, in exactly the same way. You need to process every single sample in the exactly same way. And I will show samples on that here and more about that tomorrow. Like the things that you would never have thought can have an effect on your community figure or community output. They do. And that means lots of extra use of money and time for you if you forget that beforehand. I will also hopefully be able to convince you that Ion Torrent PGM is a usable and definitely more economic alternative for the MySeq sequencing. And the last very important point is that no matter how well you do things in the wet lab, no matter how good sequencing platform you have, and no matter how well you do your after run, quality control and everything, your high throughput sequence data really never is perfect. So bear that in mind when you recover new OTUs or like novel diversity. Okay. How many of you would feel comfortable explaining at 4 o'clock in the morning what is Amplicon sequencing or like microbiome analysis? Like how convenient are you with these topics? Anna, I know you would. just raise your hand. I'm sure a lot of you. 0? Okay. Okay. Yeah. So maybe not all of you. So maybe I have something to give you during this lecture. So it's a characterization of the typical microbial community. We talk nowadays a lot about microbiome analysis. So we sequence, or by sequencing a vast amount of template sequences from just like any given community, we hope to recover a figure of the of the microbial community, like who is there and so. So what you need to remember is that this is not metagenomics. There are commercial products that call 16S sequencing metagenomics, but that's not the agreed use of the term metagenomics in the scientific fields. So it's microbiome analysis or Amplicon sequencing. Metagenomics is something that we will learn later this week. And if you analyze the microbiome, you often characterize the bacterial communities. That's what people used to do with different kind of fingerprinting methods earlier days when sequencing was more expensive. You extracted DNA and typically sequenced the 16S or 16S rRNA gene. You can also extract RNA, in which case most of your RNA will be ribosomes, and from that you can turn the RNA to DNA, and then you can sequence the 16S RNA, which has some benefits and is actually technically not much more demanding than analyzing the gene. Nowadays, there are also very much used primers that cover both bacteria and archaea. With the same primers and with the same analysis. Some people say that such primers would never work ideally for both groups, but they are used very much, and most often I think they give you what you want. And of course, it's very convenient if you can, with just one analysis, cover the entire prokaryotic diversity. You could also analyze the fungal communities. So fungi don't have a 16S, but they would have a 18S rRNA gene. Does anyone of you know or remember why we don't use 18S for fungal community analysis by Amplicon sequencing. Exactly, yeah. It's too conservative. So if you analyze just a short fragment, and actually even if you would analyze a longer fragment, it wouldn't be able to discriminate your different fungal taxa or fungal species into different OTUs. So for fungi, to recover some diversity, you actually need to use this very unconserved region, the ITS1 region is typically used here. So that is the internal transcribed spacer between the 18S rRNA gene and the 5.8S rRNA gene. So this part is transcribed, but it never makes it to ribosome. So I haven't heard, and I don't think there is any sense really to try to do fungal community analysis with ITS1 from RNA. I think the sensitivity would be very poor because you would essentially need to recover messenger RNA, and there isn't too much of that compared to ribosomal RNA. This is the figure, probably, hopefully, familiar to many of you, showing the structure of the 16S rRNA gene. So what we need to remember about that is that it's a long gene. The full length is about 1,500 nucleotides. And this secondary structure of the rRNA that you then need to use in the cell, that actually determines very much of the sequence that we have in the gene. We have conserved regions, which are very well conserved, at least within like bacteria, sometimes also with archaea. And then we have variable regions. And when we talk about the different regions, we usually refer to the variable regions that we want to sequence, because the variable sequence give us or show us the diversity. Those are the regions that vary between different genera and taxa. And the conserved regions, on the other hand, are very good places for your primers that are supposed to be as universal as possible. So the most commonly used variable regions, or most commonly sequenced variable regions, I actually think nowadays most commonly, at least Finnish laboratories, and for my sake, recommend this V3, V4 region. Some people say that that's too long. You don't get good data from that long region, so that you should focus on just V4. And actually, I think I'm under the impression that like genus or at least family level classification with just V4 works pretty fine. Earlier, people have worked a lot with the first regions, V1, V2, sometimes also V3. And for example, my data will use this one. The reason for using the first regions is that actually most of the variability in the gene is in the very beginning of the gene. But yeah, for example, if you have heard about Earth microbiome projects, and basically when you just look for sequence data in the databases, most of those cover the V4 region, and there are very good primers for that. So, Amplicon sequencing for microbiome analysis, but actually you can do so much more with Amplicon sequencing. Like when you know the tools, when you know the concepts, you are not necessarily just a microbiologist anymore, but you can expand your field to analysis of plant communities, you can recover the diversity of plants, like the current and historical diversity of plants in your field plot, by extracting DNA from soil and sequencing it with plant-based primers. You can also use it, for example, to analyze nematode communities. And you can, of course, analyze microbial communities with lots of other genes than just 16S. We have a collaborator at the University of Oulu who is interested in the diversity of antimony and arsenide metabolizing microbes in these peatland soils that treat gold mine like waste waters. And with her, we actually combined, I think, amplicons from, was it 16 or 15 different like genes and parts of genes in one sequencing run, and it was successful. So you can like mix and match lots of different things, even in the same run, and learn about diversity of specific genes and not just like microbial communities. Nitrifying communities, hydrocarbon degrading communities, whatever you choose. If you have just good primers that produce short enough amplicon. Again, a reminder, what will the microbiome data tell you? Or what do you expect it to tell you? So the really cool thing with sequencing is that from the sequences you learn identity. In the earlier days, we had all these microbial community fingerprinting methods like DGGE and LHPCR, TRFLP. By the way, how many of you have used or at least heard about of such fingerprinting methods? About half, yeah. So with those techniques, you learned how and which your samples differ from each other. But with the sequence data, you have the identities. You have just one sequence or several sequences, and you know who is this. So that's what you get. And you also hope and expect to get the relative abundances of your different community members. Just remember that it's not really the abundance of the community members, but the abundance of the genes. And next slide will show you about this more. But the abundance of these genes in each single copy of a genome varies from 1 to 15. So if you have very slow-growing, tiny environmental bacteria, they can often have just one copy of the 16S rRNA gene. And some, I think, Clostridia have some spore formers have the highest numbers, up to 15. So just remember that it's not the proportions of your cells or even the proportions of your genomes that you will get. Another important thing to remember is that even though you have your identification data, that by itself will not tell that someone really is in your sample or in your studied environment, or that someone is not there. But you need these additional things. So, for example, if you sample soil, in 1 gram of soil, you can have easily 1 billion prokaryotic cells. So 1 billion genomes. If you sample it, well, you typically extract DNA from that 1 gram or half a gram. When you sequence your sample, you get maybe, if you have 200,000 sequences from one sample, that's very nice. That's already a lot. But obviously, that 200,000 sequence, they don't cover like your entire 1 billion bacteria in that sample. So if you want to look for rare taxa, amplicon sequencing is not, like really truly rare taxa, then amplicon sequencing is not necessarily the method of your choice. Also, sometimes you will be very surprised that I found this in my sample. Like, what is it doing there? Like, why is it there? Like, this is novel finding. But again, it's not enough. You need to make sure, first of all, that the sequence really is what it seems to be, but you also need to make sure that it's not contamination. If you work with soil, like I do, you have so much of genetic material that contamination is not really a big risk, because you never anyway recover like everything that you have in your sample. But if you work with some environments with very low microbial diversity, and especially very low microbial biomass, you need to be very careful to make sure that what you find and have in your samples is not contamination. Contamination from DNA extraction kits, from PCR master mixes, those are very often, or actually almost always, a little bit contaminated. Or just contamination from your own skin, from the air in the laboratory. So just remember these when you get and analyze your data. So this slide is commercial DNA standard that I will actually come back to later on. This will just show you that if you have the same proportion of genomic DNA, really the same amount of nanograms of DNA from different species, the proportions of the 16S gene will vary a lot. So just bear this in mind. And there are actually tools to correct for this. But just bear this in mind, that what you get is not cell proportions. With 16S, of course, when you have the names tell you something. So you're not really excited to hear the names themselves, but what the names indicate. And actually they do indicate a lot. So when you hear a name of a bacterial genus, you if you are a microbiologist, you immediately think like, hey, these germs can do this process. So that's mostly what you're interested in. It's not like I have Staphylococcus aureus, but it's like I have this nasty infectious infectious microbe that can do this and this and this for these kind of organisms, or you have denitrifiers or chlorophenol degraders. So when you have the 16S data, you have indication of what functional properties or functional possibilities your community has. So you can just do research on the literature, just search for articles, and you can use tools just like PyCrust that will combine data from 16S to data from reference genome databases, and then will indicate what kind of like functional properties your community has. And why this makes sense is that according to this review from, or actually analysis from 2014, we already have pretty well recovered the microbial diversity. It's the people from the Silva database maintenance in Germany who published this. So I would argue that, and a lot of people would argue that it's the best reference database for 16S data. So if you look at the rate of species discovery, bacterial or prokaryotic species discovery, it's really going down. And this is by 2012. So rate of genera, bacterial or prokaryotic genera, the rate of discovery is really going down. And so the databases already have like so much of the diversity. Like we don't necessarily have the right names, but we have already done so much sequencing that very much of the 16S diversity already is in the databases. If you don't have sample from the moon, don't expect that 40% of your species or genera are from unknown phylum, because then I think it's just usually rubbish data. We already have like lots of, or most of the known diversity in the databases. But a counter argument to this is a publication from last year. Some of you may have heard about this candidate phyloradiation. All these unknown, uncharacterized bacterial phyla that were discovered by metagenomics, focusing on closing or making genomes from, I think this was from groundwater, like very deep groundwater that they filtered through a filter with which you normally sterilize your samples. So just tiny cells which they sequenced very deeply, and then we're able to find these new genomes and whole new bacterial phyla. So which one do I say is the truth? I don't know. Somewhere between. The nice thing with 16S-based community sequencing is that actually the results very often very well align with metagenomics sequencing. The price difference for the two methods is huge. But for example, in this publication by the Ferrer group from 2012, when they did the unconstrained ordination for samples from very different kind of biomes, you can see that the results, like which communities or which samples resemble most each other, which cluster closest together. This figure, based on 16S rRNA and based on metagenomes, it's almost the same. Also, when you look at diversity, 16S diversity and functional diversity, also that in this article they found that the results based on 16S and metagenomes, they give those results correlate really well with each other. Okay, so that was background or theory. Then we'll go to more of the things and recommendations on like what do and what definitely don't do. So the effect of PCR conditions. These examples are mostly actually from my background with community fingerprinting. When we use those, some people would nowadays say that those are outdated methods because sequencing will give you so much more data and better results. Well, with sequencing, you definitely, or most people would expect them to differentiate your samples better because there's just more data that you get out of the analysis. So if you expect better separation power, you should definitely expect like more separation power due to like unwanted things as well. So even though what I show here is results mostly from community fingerprinting, like you could very well expect that differences would be even exaggerated or enhanced by doing the same analysis based on sequencing. So back to the wet lab. Just remember that everything, like every single small choice that you do when you set up the PCR or when somebody sets up the PCR, the amplification of the gene fragments for you, like everything impacts the outcome. So for example, if you use a little bit different annealing temperature where the primers anneal to your template, that has an effect on the recovered community. If you use different master mixes with different magnesium chloride concentrations, you will recover a slightly different community. So what you see here is that when you have less stringent annealing conditions, so when you either have higher magnesium chloride concentration or lower PCR annealing temperature, so less stringent conditions, you recover this whole new group here that you don't see in the more stringent conditions at all. Does anyone have an idea why that could be?
Primaries priming, it's not exact match, but it's priming something that's almost match.
Yeah, exactly. So actually, we call our primers universal primers, but they are not necessarily truly universal. So you can easily have like 1 or 2 base mismatches, and you expect your primers to work if you have 1 mismatch. But if you have 1 mismatch, the annealing temperature will be lower. So you will have bias if you use very high temperatures. You will lose those community members because the ones to which the primer anneals or matches perfectly are amplified much more efficiently. So based on this kind of data, I would recommend that use the lowest priming temperature or annealing temperature that you can, because then you have the best chance of getting all of your diversity. What of course limits this is that you don't get too many non-target sequences. So for example, if you try to amplify just bacteria, that you don't get archaea. Usually that's not really a big problem. And you can anyway later on filter out the archaeal sequences. Another problem, like a technical thing, is that at some point you will start to get more primer dimers. But I was actually recently, or I have been surprised by how small a problem that is if you have a good amount of template DNA. By the way, so you will get all these slides, so no need to write everything down. We will put them online later on. Another thing, the polymerase or master mix. In one of our earlier works, where we wanted to recover bacterial and archaeal communities from the very same samples, based on both RNA and DNA, and then make comparisons. One of the problems, or one of the challenges, was that we never got the archaeal primers to work, or the archaea to amplify, when we used a proofreading polymerase that didn't make so many mistakes and was supposed to detect the targets more specifically. So using this fusion polymerase that maybe some of you have also used, it never worked for archaea. We tested everything, but then we had to do it with the regular Taq polymerase. And of course, when later on cloning and sequencing our archaeal amplicons, we were a little bit thinking like, what will we get? Like, is it archaea at all? But it was all archaea. But it was just that the other polymerase didn't work. So these kind of things you may see. Taq polymerases may work better. And actually, a lot of people use Taq polymerase. I spent 10 months at Noah Ferrer's group at the University of Colorado in Boulder, which is probably one of the biggest or well-known groups doing environmental microbiome analysis. And for example, in that laboratory, they use stock polymerases, or they actually use a ready-made commercial master mix. And then when one commercial master mix didn't work well, or there were problems with delivery, then they just changed to another commercial stock-based master mix. So I think that's what people mostly do. A lot of, I think, Finnish sequencing laboratories recommend to use proofreading polymerase. And there's definitely no harm if it works well on your samples, if you got your targets to amplify. But it will cost a lot more. And the counter-argument to use stock polymerases is that even though you have errors in your data, the errors introduced by PCR will be anyway so much less than the error introduced by sequencing. So it's kind of like you don't even see the PCR error. That's what some people expect. Another thing that people usually don't take into account is template concentration. So when you do your very first PCR with those general bacterial or archaeal or fungal primers, especially with the prokaryotic primers, I think it's a fantastic idea to quantify your extracted DNA concentration. Also because that is actually a very good estimate for biomass. Usually the biomass and extracted DNA concentrations correlate beautifully, actually perfectly. But another reason is that, if you only can, please put the same amount of template in all your reactions. And the reason you see here. This was, we had two water samples, water sample 1. This was the DNA concentration in the extract, so about 0.05 nanograms. And then we had a second water sample where the DNA extract concentration was about 0.5 nanograms per microliter. So yeah, at the lower end, but definitely not ridiculously low DNA concentrations. And then you put the same amount of microliters of DNA extract, or its 10-fold or 100-fold dilution as template in the first PCR. And then you continued everything else as you usually do, and then you get the sequencing results. And here we have the relative abundances, actually in this as a percentage? No. Well, the relative abundances. Yeah, the relative abundances of different bacterial phyla. And you can see that for actinobacteria, it almost halves when you dilute your DNA sample. In both samples. Here it actually reduces to about 1/3. For Bacteroidetes, it also, in both samples, you see less sequences when you dilute your sample. And for Proteobacteria, definitely much more sequences, or much bigger relative abundance, like here from 37 to 66%, just because of different template concentration. So this is not talked very much, and I don't think there are very many publications about this. But if you can make sure that you have the same amount of template, then I think you are on much safer ground when you interpret your sequencing results.
Can I comment from the human side?
Yeah.
I do where we see the complete opposite. Where because of inhibitors in the PCR, so that the more you dilute, the better the composition is. You can get more of certain species, for instance, exactly Amplicon are difficult with the primers that we're using are difficult to amplify. We see higher abundance with the it's titrating how much you dilute, but then it also works the other way around, that if you dilute, you get better
So how do you then make sure that you get the same, that it's the right? Do you dilute as?
We the amount of template that we put for sequencing.
Yeah.
So basically, do you use nanodrop or do you use better, more sophisticated methods of measuring the DNA quantity?
Well, at least for environmental samples, you need to use these like picogreen based specifically DNA binding. But how about how do you have human DNA? Will that interfere in your DNA quantification?
Well, yes, and that makes it's a whole other ballpark. If you have biopsies where of biopsy samples, in fecal samples, it's the human contamination is not a problem.
Okay.
There's so much more bacteria than human cells.
Yeah.
And the way that we extract kind of gets reduced. The human cells are broken. So in the beginning phase. So but it is a problem, especially in the biopsies where you don't know. Yeah. You have DNA, but you don't know where is it from.
Yeah, it's the same when we analyze plant roots or when we want the entire root community, also the endophytes living within the root. So then we just freeze dry and break the entire sample, and then most of it is plant DNA. What you could then do is quantify the amount of your prokaryotic cells with qPCR, and that.
We do next because we biopsies we have so little sample that we might do nested PCR. So we not by the whole of 60 nests and then do a small, the next whole of the PCR bites.
We do the same actually with the root communities. Yeah.
Have you ever checked whether the different proportions of eukaryotic DNA as such affect the results? So if you have highly varying proportions of eukaryotic and prokaryotic DNA in the samples,
no.
Probably as this theory might have.
It could. For example, if we just with our normal prokaryotic or bacterial protocol analyze these root samples, actually what we got with this one species, plant species that we were working on, we got 95% of the sequences was the mitochondria of that root. So yeah, what was left was of bacterial sequences was obviously way too little to make it like economically sensible, but also it might have been biased. So we ended up with using this first these primers that discriminate for the mitochondria, and then continued with the normal protocol. But yeah, everything affects. Also, sequencing platforms affect your result. So here are some earlier references comparing MySeq data to other sequencing platforms. Nowadays, I was at the soil metagenomics meeting in Germany, Braunschweig, in December, and heard a lot of very useful tricks there, what is really not just published results, but there were a lot of presentations and workshops and what is done nowadays and what things cost and what is recommended and what is definitely not recommended. There you had a lot of people saying that we use MySeq, don't use anything else except MySeq, MySeq is good. and definitely don't use Ion Torrent PGM because the data quality is good enough. And they base those comments on these two earlier publications that are actually very nice publications. So here you see that the Ion Torrent data quality is much more than half worse, or the error percentage for MySeq is around 0.5, and here for Ion Torrent it was closer to 1.5. And also here error sequencing in this another publication. One thing that you should definitely bear in mind is that don't try to merge OTU or sequence data from different platforms, because the data that you get is just very different. It has different kind of error profiles, different sequences. If you really need to compare data from different sequencing platforms or sequencing chemistries, it's better to turn those into relative abundances of genera or families, and then it's much more safe to compare. But like, don't try to merge OTUs or sequences. Actually, don't try to merge them even between MySeq and HiSeq. When I was in the Fierer lab, they were just wrapping up this 1,000 homes comparison. So they had collected dust samples, or actually put the inhabitants to collect the dust samples themselves from outdoor door, I think it's called door rim, but anyway, what's like above your door, and indoor door rim. So the outdoor microbiome and indoor microbiome. And they did most of the sequencing with HiSeq, because when you have 2,000 samples, like, that's the sensible thing to do economically. Then they got some new homes. They wanted to redo some samples, and those data they sequenced with MySeq, with which you normally do your microbial community analysis. And what they were just like, what was one of the big issues that was talked about when I was there is that you couldn't combine the data. They realized that the MySeq data and the HiSeq data clustered separately. So I don't have like a data to show you about this, but just that people have tried and it doesn't seem to work very well. Not a good idea. Also, the ion torrent has different chemistries. It has developed a lot. So, for example, so these reports are with chemistries of ion torrent that I would call completely outdated, or very the early versions. And I would definitely not merge like current data with that kind of data. But yeah, because we have ion torrent at University of Jyväskylä, and we use that. And actually we have so far never got comment from reviewer that this is bullshit, like, why didn't you use MySeq? So you can do it, no worries. But when you go to conferences, it's kind of annoying that everybody tells you, like in the presentations, that, you know, just use MySeq, like ion torrent doesn't work. So I wanted to see, like, what is the quality with the current chemistries, with the most recent versions. Because I have seen it with my own eyes that it has gotten much better. And I don't actually have the comparison of the different versions. But what I did was that I took the very same sample and sequenced 4 replicates or 4 barcodes with our in-house ion torrent PGM. And then 4 replicates were prepared with the MySeq barcodes and sent out for sequencing at the University of Helsinki Institute of Biotechnology. So these are the specs from the most reagent kits. MySeq 2 times 300 base pairs, and the price that we got, I think it also always depends a little bit like what you negotiate. I don't think you will get your sequencing at much cheaper price than this. Here I prepared everything myself, and yeah. For the ion torrent, our in-house protocol, just for the sequencing, the cost of reagents is about €7 per sample, just for the reagents, without what's called tax value, yeah, without VAT. So a big difference in price, and how much time do I need to invest in the sequencing? If I do the sequencing myself for about 100 samples, it takes about, the sequencing itself takes about 1 day. So not too much. I used the exact same defined template that had just known sequences. So it was this mock community consisting of known strains, or genomic DNA from known strains. And our in-house protocol is for the variable regions 1 and 2, where you have a little bit over 300 base pair Amplicon. So it should work very nicely with both chemistries. Actually, the protocols nowadays recommended at the Institute of Biotechnology and for example by the Turku sequencing facility that a lot of people nowadays in Finland use is V3, V4 protocol that will give you a longer amplicon than this. So this was like a nice small amplicon that should work pretty well with both. Commercial DNA standard, because I wanted to have some microbial diversity there. I definitely wanted to have genomic DNA and not just 16S amplicons that I would have pulled. And we had gram positives and gram negatives there, bacteria with very different GC content, which goes from 66 to 33. Different numbers, of course, and also somewhat different versions of 16S, very different genome sizes. But yeah, it's genomic DNA. And the nice thing is that the full 16S rRNA gene sequences are available online for the different copies. So these are fully sequenced strains. So you know what you should get, which is essential if you want to do error profiling. So just to make sure that sequencing was really the only difference, I did everything by myself. So I used the same amount or same amount of nanograms, 5 nanograms of template per reaction in the first PCR. And I did the first PCR in triplicate, which were then pooled and then divided for the different barcodes in the second PCR. So the first PCR just amplifying the target region. For PGM, it was just the general primers, and the MySeq primers had already overhangs at both ends, these partial TrueSeq adapter sequences. But as you can see, these are the PGM amplicons. This is, by the way, a figure from qPCR, so you see how your templates amplify. So they come up a little bit earlier than the MySeq templates, but I would say that they amplify. The reaction has worked just as beautifully for both. So the adapter sequences or the overhangs didn't seem to like really impact or had a negative impact on the first PCR. In the second PCR, if you do this like two-step PCR protocol, in that you add your sequencing adapters and barcodes. So that I did using the pooled product of the first PCR as template. And 4 barcodes per platform. By the way, this is one really convenient and nice thing to do to use qPCR master mix, especially in the amplification of the first template, in the first PCR. One point is that you don't necessarily need to run a gel. When you have like a known protocol, and you have worked with the primers before, you know they should work, and there shouldn't be a very big problem with primer dimers or anything. So when I see this kind of amplification result, I don't bother nowadays ever to run a gel, because it works fine. Another thing is that if you put in the same amount of template, you would hope your samples to come up, to start to amplify at about the same time, which is what you would want to see. And another thing is that when you do this, you can see like how the run develops, and you can stop your reaction at the early phase or even beginning of the plateau, which would be ideal because then you would have less heteroduplexes and probably less chimeras, which are problems that you introduce, or errors that you introduce upon PCR. Actually, it would have been ideal to stop it even here at 20 cycles, but yeah, I had some issues. But still, you see that the plateau PCR hasn't been at plateau for a very long time. Also here, you could have stopped it at the beginning of the plateau. Just like a trick to use a little bit more money but save a lot of time. So again, I purified the 4 replicate barcodes for both PGM templates or PCR product, PGM PCR products and MySeq second PCR products. I purified them with the Ampure purification system, and then I pooled the 4 replicate barcodes together, and then I checked with this TapeStation, which is like a newer version of bioanalyzer, a very sensitive automated gel running system. I checked the quality of pool, and as you can see, I think the quality of the pool is really comparable. You have some crap here for both, but actually it's a very, very good quality template. So just to make sure that you have the same quality template going to both sequencings. Then I use and like the mothur processing pipeline a lot. So I used that. And I used the mothur standard operating procedures, or SOPs, for 454 data, for the ion torrent data, and the MySeq SOP for MySeq data. So just that, not something that I invented myself, but like an established procedure. The only difference to that was that I didn't use sequence-based quality filtering for the ion torrent data. And I will tell you later why I didn't do that. But in essence, only the really beginning of the pipeline differs. Otherwise, it's an identical pipeline. So I use the Silva database for alignment and classification, which, according to Pat Slosh, the maker of mothur, is the best one. I did chimera detection and removal with a U-chime that is used in the 454 SOP. Here, I think in the SOP they use a different tool, but I thought that I should use the same. And I did clustering with OptiClust, which is the newest clustering method. And then if you want to see how I did the error analysis, you can find the definition in this 454 SOP. There they do what I did. Just a couple of commands. I can also give you the full, like, all the commands that I use, because yeah, there is like nothing new or nothing strange. So if you want just like a text file, just ask me. So error analysis per sequence shows you how big percentage of sequences has 0 errors, 1 error, 2 errors, 3 errors, and so on. And then these are these combined categories. So MySeq and PGM. There is a small difference in most classes for the favor of MySeq, But based on this, I think it looks pretty good. I think this pretty well justified me using PGM. Of course, you don't see these categories very well. It doesn't seem like there is anything there, but actually there is. So when you put this scale as logarithmic, you see that actually there are not a big percentage, but anyway, significant amount of sequences that have lots of errors with both sequencing platforms. The error analysis, a number of sequences. When you calculate it based on the 454 SOP, for MySeq, the total error is 0.2, and for PGM, it's a little bit more than double. So yes, there is still a significant quality difference. Another thing is that for MySeq, I'm not actually sure if I got the really typical amount of sequencing or was it a small run. But I got lots of sequences, above 150,000 or per each of the 4 barcodes. And because I was able to pool them at very equal proportions, the difference between the highest and lowest sequence count per barcode is not very different. For Ion Torrent, I got more than 25,000 sequences per barcode. And yeah, these are the final good quality, filtered, classified bacterial sequences. The amount of unique sequences is huge. And for both of the techniques. It actually seems to scale up with the number of sequences. So I think the amount of templates, well, it was 8 different bacterial species, and some of them had different 16S copies. So I think the total number of templates different templates was 10 or 11. But still, this is the amount of unique sequences that you get. Lots. So sequencing error and also PCR error. Also, you can calculate based on the error checking protocol. You can check how many true chimeras there are that the U-chime didn't detect. So here you can see that. Yeah. Yeah.
How many singletons you have? If you leave out the singletons, what's the amount of unique?
In the mothur protocol, I did leave the singletons, but I leaved out singleton OTUs and not singleton sequences, because I'm not sure if there is a command. Does anyone know if there is a command in mothur? Oh, okay. Oh yeah, that's true. That should work. Yeah. So I actually don't do that for sequences. I do it for OTUs. It's a little bit more conservative to do it on OTUs. Singletons should be justified as well. For example, in Vierer Lab, they do it on sequences. Yeah. But we just got reviewer comments saying that you should never leave out singletons. There is no... It's like... Which I don't agree. At least for OTUs, I think it's a great idea to leave out singletons because, like, you always have replication. If you don't, like, if it's just like single observation, no, it doesn't really tell anything. But yeah, much more sequences for MySeq than for PGM. Lower error rate, but I think, like, I'm pretty happy with this error rate. If I need to pay €7 versus 50 or 60 euros, I'm fine with this. Especially when the sequencing in-house I can do in 1 day, and if I send my samples out, I usually have to wait for 1 month, but or more. It depends a lot. Then of course the big question is how many sequences do you need? Like, if your supervisor doesn't know anything about anything, he or she would probably say that, of course we want 2,000 sequences per sample and not 20,000 sequences per sample. Like, I don't know, how many of you have heard this comment? You've probably read it from, like, these quotes, at least. The fact is that when you compare alpha and beta diversity patterns, so when you compare the diversity of the microbiomes, or when you compare how well you find the patterns, like which samples resemble each other and which like less and which resemble each other more, actually the patterns are usually very similar with 1,000 and 100,000 sequences. You can test it when you have your own data. You can verify your data to different level of sequences, and at least what I have seen and read so far is that usually you have very much the same patterns. Another question is that what are your computational resources? So if you don't have an in-house supercluster, we actually, by the way, what we do this with is that we use the PGM server, because the sequencing machine comes with a 10,000 euro computer, and when it's not doing sequencing, it's an Ubuntu, like very nice Ubuntu a cluster with an Ubuntu or Linux operating system. So we actually run mothur with that. If we wouldn't have that, I would definitely all the time be using CSC services. I would not try to run things on my own laptop anymore. I did that 2 years ago, 3 years ago. If you don't like thinking about the computational resources, it may actually be nicer to have 25,000 sequences than 200,000 sequences per barcode, especially when you think about the number of chimeras and unique sequences seeming to scale up with the amount of sequences. Another point is that if you are very interested in some of the rare taxa, rare families, rare genera, you know that they are not very highly abundant in your sample, in the bacterial communities of your sample, but you still want to detect them and maybe even do some statistics on them, then you would want lots of sequences. For OTUs, so from which I already filtered out the singleton OTUs, so OTUs that had just one sequence in that operational taxonomic unit. By the way, do you know what is OTU, or operational taxonomic unit? Who feels a little bit, who wouldn't be comfortable explaining it at 4 o'clock at night? Many of you. So OTU is operational taxonomic unit. In bioinformatics, it's a collection of sequences that don't differ from each other more than typically 3%. So you have a collection of 97% identical sequences. You can use other thresholds, 5%, 1% different, but almost always it's this maximum 3% difference. Why you use this is that, well, we already heard and learned that singleton sequences or unique sequences are crap. There is sequencing error, there is PCR error. So you shouldn't worry about singleton like unique sequences. Another thing is that it somewhat correlates with the idea of bacterial species. So it has been said that bacterial species for full-length 16S on average differ from each other 3%. So if you have 4% different full-length 16S, then you could consider it as a new species, like based on this rule of thumb. So, operational taxonomic units, and we will be talking a lot about these later this afternoon, or later today. So actually, even though we had more unique sequences, we had less OTUs for MySeq. And we found exactly what we wanted. So all 8 genera were found in 8 most abundant OTUs. And the ratios were relatively even. Why we have so many OTUs and not about 10 or 11, how many unique sequences there were? Or actually probably we should have 8 OTUs, because the different copies of 16S don't differ that much within the same species. Is that a better merge tool in the pipeline could maybe reduce this problem. For PGM, we actually had much more OTUs, which may be because for PGM, the typical error is a homopolymer error. So when you have several copies of the same nucleotide, PGM is not as good dealing with that kind of regions. So that's the error for PGM, and apparently that leads to seeing more OTUs. For MySeq, I think it's just like a single. The main error is just you either have a single nucleotide difference. So for a single nucleotide, it's not able to say whether, like which base it really is. But anyway, also for the PGM, all of the different genera were found in 10 most abundant OTUs. But Bacillus was split to 2 OTUs, and Lactobacillus, even though present, it seemed to be underrepresented. So I need to find out like why is that. And one of the reasons was that apparently some of the Lactobacillus sequences were terminated prematurely. So if the sequence minimum length at the very beginning of the sequence processing pipeline was changed from 200 to 100, then I got what I wanted. So all genera found in 8 most abundant OTUs, and classification to the genus level, which I think you should and could expect with Amplicon sequencing. Don't expect species level identification with Amplicon sequencing, because species level identification works on full length 16S and not really well with the shorter fragments. With PGM, maybe you remember in the beginning that I didn't do any quality filtering. And doing that could reduce this issue. I'm still working on like what would be the best process for quality filtering, and I will come back to this a little bit later. If you really want to compare sequencing platforms, I don't think you should do it just based on one analysis pipeline. So I did the whole thing. I analyzed all the sequences also with a different pipeline, a commercial one. There's a program, a pretty expensive but also a very user-friendly one by CLC Bio. It's nowadays owned by QIAGEN. And if you want to test it out, it's I know a lot of people like use it for other things. Your institute may have it already, and this OTU clustering pipeline is a pretty new thing that they have. So if you want to test it out, check if your institute already has it. If not, but you are really interested, you can get a, I think, 30-day free trial license, and it won't take you more than 1 day to learn to use it. So you can do lots in 30 days. And yeah, it's very efficient and very user-friendly. But it's expensive. And you don't have full control of everything as you have in the command line. But yeah, I processed both MySeq and PGM data with exactly the same way. So the only difference was that for the MySeq data, the two reads from different directions, R1 and R2, were merged before the first quality trimming. And I used the default parameters. So there are quality trimming parameters. I used the default versions. Trimming to fixed length based on automatic determination of the length. And I used a fully reference-based OTU clustering. So I didn't allow it to detect or identify novel OTUs, but I told it that, you know, because it's these known microbes that are very well represented in databases. So why not do reference-based OTU clustering? I know that my sequences are in the Silva database, so just look against the database and no novel OTUs. Classification results for MySeq. The 1, 2, 3, 4, 5, 6, 7 most abundant OTUs, or the main OTUs, had 7 of the right genera. But for some curious reason, Pseudomonas aeruginosa, that should be, this is the theoretical composition of the 16S counts. Pseudomonas aeruginosa was missing from the main OTUs. For PGM, also 7 of the main OTUs were there. There was something very funny happening with false identification of Acinetobacter. And yeah, I got this data at the late last week, so I haven't had time to like see everything. I got the MySeq data on Wednesday. So I need to get a look into what is this. But apparently there is something funny going on with the pipeline. Maybe with chimera identification. That's my best guess. That for some reason it identified some sequences as chimeras or some chimeras as sequences. And, also here we have one genera or one species missing. So this Lactobacillus fermentum was missing. Okay. Yeah.
How many reads do you have per sample based on before doing the filtering?
Let me think. For PGM, you typically lose something like, if you don't do quality filtering, you lose maybe 20%. For MySeq, I think even less. So I didn't lose like, you know, sometimes you lose 90%. So not that much now. I can give you, I actually wrote all these down, like what I lost in which step with my protocol or with the commands. So I can give you that, and there you see all the data. But yeah, so there are lots and lots of different analysis platforms that will give you some different results. Many of the names are written or pronounced in a really strange or funny way. I don't think it makes any sense that mother actually, so this is spelled mother, not mothur or something. And also it's usually written with a small, like not with capital M, but with small m. This is pronounced chyme, which again doesn't make any sense, but that's the correct pronunciation, chyme. And this one, which you nowadays use in like together with chyme, it's like this online database and tool. This is pronounced as cheetah, as the animal is gepardi. And yeah, it's it's just nice to know that they are pronounced, but it really doesn't make any sense, and I think it's horrible annoying to do, like, give names like this that are spelled, like for a Finn, like, you know, spell the things as they are pronounced, like that's the proper way to do it. Then there's USearch that a lot of people use, and for example was used very much in the Fierer lab, and then commercial versions like CLC. So then the question, like, what to use and with which parameters? So I will just like tell you briefly about some known issues, like some like known problems for which you will also find references and articles and definitely lots of like online discussions discussing these. But just like beware, like it's good to know a little bit. For MySeq data, one of the big challenges actually nowadays seems to be like merging the two reads, like this very elemental first step that should work automatically apparently doesn't. So if you, for example, go to check this USearch manual or just read publications and or discussions, you will find that USearch seems to work best for merging R1 and R2. Especially if you after that do quality filtering based on expected errors, so using max EE. And for example, at the Fierer lab, we used max EE 0.5, which is very stringent. At the USearch manual, I think they recommend max EE 1, which is still pretty like tough or stringent. If you can do that and still have enough data, fantastic. If you sequence anything else than just V4, you probably will not have enough data after that. So if you have full overlap of R1 and R2, so if you sequence just V4, any of the tools should work fine. But few of Finnish laboratories or few people nowadays like are happy with just V4. You get more greedy. You want at least V3 and V4. If you want to have really good quality data and want to do it with MySeq, then just sequencing V4 would really be a great idea. So probably much of the unique sequences or funny things that we saw in the MySeq data were actually because of the original merge and quality filtering that is embedded in the merge tool did not work perfectly. And Patch Loss, the maker of Mothur, he tells it all the time, like, yes, it doesn't work, like you should have full overlap. It doesn't, like he fully admits that the merge tool in Mothur doesn't work very well if you don't have full overlap. Another thing that you can read about both in blogs on different pages is that the newest version of the MySeq kit is definitely not better than the previous version. So especially the R2, the reverse reads seem to be actually worse than from the previous version. But I don't think you can order the previous version anymore for MySeq. And based on some initial tests of mine, the CLC pipeline seems to work pretty well for the merge and for the quality filtering. But it's not free. And also for USearch, the multi-core version. If you have more than 20 samples or 10 samples, you need to use the multi-core version, and also that is not free. You need to buy the license. For PGM reads, you have just one forward read, and always in sequencing, in Sanger sequencing, in Ion Torrent and PGM 454, like always in sequencing, your quality drops towards the end of the sequence. So when you get Torrent data, you always take it through this Torrent suite or server. And it already does some initial quality trimming for you. So it starts to check from the end of the sequence when the quality gets good enough or acceptably good. And then it starts the actual sequence that it gives to you. If there are poorer quality regions, like it first goes up and then it drops again, it still thinks it's an acceptable sequence. So poorer quality regions, like within the sequence, not at the end, those are an issue. And I found, and you will find, publications that have used this quality window filtering in mothur for MySeq data. Several publications used a quality window of 10 base pairs and minimum average quality of maybe 25 or so. Yes, you can do that. You get better quality sequences, but you will also introduce systematic bias. So with the latest sequencing version, I noticed with the latest sequencing chemistry that gives already pretty nice quality data without any additional quality filtering. I noticed that when I did the pipeline without initial quality window filtering and without it and with it, I had significantly different proportions or different amount of sequences of certain genera. So there was definitely a bias, a discrimination. Especially for nitrospira, I think that the number of sequences reduced, like not just the proportion, but number of sequences reduced was like 300. Now I don't remember. Well, maybe something like 80% difference. Anyway, big discrimination. So it has been published, it has been used. I would not recommend that. And also I think probably the issue was the same for 454 data because it has the same kind of error profiles. So quality window filtering, at least with small quality window, probably caused some systematic bias in that data as well. How much do I have?
May I ask one question about
Yeah.
the previous topic, so the merging of leads?
Yeah.
So if we have V3, V4 data,
yeah.
we don't have these commercial better version tools. Would you even recommend using only forward or reverse leads?
That was actually what was done at the Fierer lab for the ITS sequences. So for the ITS sequences, at that time we had sorry? Of fungal community profiling based on the ITS1 region. So there, because we knew that the length variation was huge, we expected that if you try to merge them, then that will cause discrimination because the longer sequences will not merge well. So there we just checked which one was better, and of course R1 was still at that time almost always better, and just use that. So you can do that also if you are worried about creating taxonomic bias with.
But also from experience.
Yeah, I think if you try to amplify like too long region, but I'm not actually sure. For example, for ion Torrent BGM, the problem is not like the sequencing. You will never get like most of your sequences 400 base pair long, even though that's what they say. But not most of them will never be that long. But the emulsion PCR, the first step in the preparation of the sequencing template, will work for that long and a little bit longer. I'm not actually sure in MySeq. Like, I think you can there sequence as long as your template can be as long as you want, but you will just only sequence as long as your chemistry says. There doesn't need to be any overlap. I'm under the impression that in MySeq you could have like 600 base pair long templates. But merging would be then the discriminating or the problematic issue. But yeah, using just read one, perfectly fine. Again, lots of publications with that done. Yeah.
Related the users to there's nowadays available this research tool. Do
Yeah.
you have any information in this case or is it
I think it's the same, but I haven't. Yeah, I think the command is the same, but I haven't used it. Okay, one last thing, or one big known issue in the field is OTU clustering. So when you cluster your sequences into these groups within which sequences typically differ from each other not more than 3%. OTU splitting, what you already saw in my data, or OTU count inflation, is definitely a known issue with actually all algorithms. And according to USearch manual and people's comments and tests, and I think also publications, USearch again seems to perform best when you read the data that is already there. And again, the multi-core version is not free, and that is actually an issue because I think this merge is something you can do, like probably even using the single core version of USearch, because for the merging you could process your data in like different batches. You could take 10 sequences, next 10 sequences, next 10 sequences, and merge them. That shouldn't be a problem. So actually that's a way to bypass the paying. But for OTU clustering, you really should do that for your entire data set at once. So there you couldn't use the single core version of USearch. But in the most recent version of Mothur from this year, they have implemented a new clustering tool called OptiClust that, at least according to the publication, works really nicely. And also in my experience, like based on the short experience that I now have on it, it seems to give less OTUs than the older clustering algorithms. And what is really nice that it's really fast. It requires less memory and it's really fast. I think the time usage was something like 5 or 10% of what the previous algorithm required. Because if you have lots of data, this OTU clustering is the like what will stop you. Like it requires lots of memory, lots of cores, lots of time in any case. So this clustering algorithm, I definitely recommend to test it out. It seems to be working well. And check out the publication. In the publication, they say that it works. Was it as well or better than USearch? So it's cool that we have the course now and not like last autumn when we didn't have this yet. Again, the same take-home messages. So I hope I managed to convince you to think about this. So if you don't know exactly what you're doing and why you would change things, then just do what everybody else is doing. And really remember that everything you do affects the recovered community profiles and relative abundances. So process all your samples in exactly the same way. And seeing these comparison data, I at least am still fully happy using Ion Torrent PGM. If you want to use it commercially, well, we collaborate with people, but we cannot collaborate with everyone. But University of Oulu sells PGM runs at a definitely price that like, of course they ask more than just the consumables, but it's still definitely cheap compared to MySeq sequencing. And yeah, high throughput data is never perfect, so beware when you make claims about new discoveries or novel diversity or yeah. So more questions, comments, discussion?
There are no questions.
Or,
I was just thinking, have people tried with like standardized test communities where there's much more taxa than what have been used in?
yes, I think yes. So for example, those comparisons, for example, that comparison or testing of the OptiClust, you can also have like in silico mock communities. So yeah, you just take sequences and then you insert some sequencing errors and yeah, 8 is very little. I think usually people use maybe like 10, 20. Yeah.
But in a typical natural community, how many
Yeah,
taxa do
yeah,
you
that's
get compared to that?
yeah. In a typical soil community, I would expect like real diversity to be, I don't know, 100,000 species, 10,000 species.
Yeah, then you can just
No, not many that, no, not with that many. Yeah. It really depends on your sample, but usually it's hundreds or, yeah, or much more. Yeah. I'm within time. I took much more time than I expected. 1 minute.
So if there are no more questions, then let's end.